We're moving into lab 3. So we have the lab manual, and going along with that, there are some images here of the cell. And here we are, lab 3. So this first section here is truly just this textbook figure, and we'll go through the details. So let's go ahead and bring this up here. So the image is labeled for us already here in the textbook, and we'll just point out the details. So we can start with a structure, a molecule known as a phospholipid. And the phospholipids here are illustrated as the red circles with the black tails. So I just need to find that label somewhere. The red circle of the phospholipid molecule, so that's the hydrophilic head, and the black tail of the phospholipid, that's the hydrophobic tail. But all together, that is one phospholipid molecule in the cell membrane. And let's just go back to the list. We have the peripheral proteins and integral proteins. Peripheral proteins, these are proteins that are found just to one side of the cell membrane. So that's what's meant by peripheral. And integral proteins pass completely through from one side all the way to the other, completely passing through the phospholipid bilayer. That is a integral protein of some type. There's the cholesterol. This sits within the area of the hydrophobic tails, and they're illustrated here in yellow, so representing the cholesterol. And we see just on the inside of the cell membrane, we have here representing the cytoskeleton. So these protein elements here just on the inner surface of the cell membrane. And we have a glycolipid and glycoproteins. So the glycogen portion, the sugar portion, that's represented here in purple. And we can see that we have the sugar structure, carbohydrate structure connected to the protein, making this a glycoprotein. Whereas over here, we have a glycolipid. So again, we have the sugar portion, and it would be attached on to a similar type of hydrophobic tail structure, but we can't clearly see that here on the illustration, but we do see the labeling for glycolipids. So truly just knowing the structures on this feature here. And now the last part for this lab, there's a figure of a textbook, a figure of a cell in the textbook, and also there's the model. So just simply naming these organelles. So we'll start with the nucleus, mitochondria, the Golgi apparatus. In chapter 2, we have this textbook figure, and of course here it is labeled. So I'll let you go ahead and just study this. And we have here is a photograph of the model that we'll use as well. And keep in mind there already is an answer key for this model, and I'll go through it as well. So the nucleus, the mitochondria, the Golgi apparatus, those first few elements. So it's this entire feature that we see here is the nucleus. We don't need any of the detailed parts. The mitochondria are all seen here in the orange and brown color. So there's multiple mitochondria. The Golgi apparatus seen here in pink and blue. So these features here making up the Golgi apparatus, also known as Golgi complex is another term for it. And then I think next after that, we probably need what, the centriole. Yeah, there it is. Centriole and the two types of endoplasmic reticula. So the centriole is this complex seen here. We have the entire centrosome, and the two yellow tubule structures specifically are the centrioles within the area of the centrosome. For the endoplasmic reticulum, we have right here, you notice, the labeling of smooth endoplasmic reticulum. smooth endoplasmic reticulum because it lacks any ribosomes. The ribosomes are represented here as all these white dots all around. So down here is a good representation now of a rough endoplasmic reticulum that contains the ribosomes in white. Smooth endoplasmic reticula lacks ribosomes. And that would be the next term. The white dots are the ribosomes. And microvilli are not seen on this model. So that's one of the reasons we need this textbook figure. And we can see the labeling here. These numerous shorter extensions are the microvilli. And again, all those other same parts quickly. Smooth endoplasmic reticulum. We could see, of course, the Golgi apparatus. We have represented in this case in blue, the multiple mitochondria is the plural form, mitochondrion singular. We should be able to see the area of the rough endoplasmic reticulum with the ribosomes attached. And in this case, the black dots are the ribosomes. We have the area of the two centrioles, again, located in the centrosome is the term. The nucleus as a whole. So just studying these three images, knowing the features of cell membrane, and the organelles on the illustration and on the photograph of the model. And that's all of Lab 3.